Hello again, everyone. So, what I'd like to do now is just continue on our discussion with regard to chromosomal mutations or chromosomal abnormalities. So far, we've talked about aneuploids and we've talked about one type of polyploid. So, again, the aneuploids are conditions where you deviate from a normal state with regard to genome organization. We either have extra copies of chromosomes or you're missing one or more chromosomes from the normal genome. So, in the case of autopolyploids are euploids. And what they are is, again, they deviate from the normal genomic state in terms of chromosome number by having extra haploid sets of chromosomes. And those extra haploid sets are derived from within the species. So again, that the way of generating these autopolyploids is shown in this diagram here, where again, if the normal chromosome state or genomic state for the organism, this hypothetical organism, is diploid with a 2n number of 6. Here you have a triploid configuration with 3 haploid sets of chromosomes, a total of 9. And here you have, again, a total of 12 chromosomes. So again, another autopolyploid derivative that now contains 4 haploid sets of chromosomes that define that particular organism or that particular species. So we've talked about that already. So what I want to do is contrast it with another type of polyploid that we call an allopolyploid that has a slightly different mechanism of production. So again, the way that I've described this previously, I've shown you this diagram before, is the idea that the end result of creating an allopolyploid derivative is an interspecific hybridization event between two distinct but closely related species that have different genomes, different chromosome constitutions. Okay, but it's actually a two-step mechanism. You can actually combine those two diploid genomes into one new genome. And in doing so, you've created a fertile allopolyploid derivative. So again, through an interspecific hybridization event and a two-step process, you can combine two original diploid genomes from two original progenitor species to combine them into the allopolyploid genome, which now represents a new species. And again, it would be a new species because it could not, the allopolyploid derivative couldn't intermate with either of the two original progenitor species and produce fertile offspring. So what are the details of this two-step process to create the allopolyploid derivative? So again, as I said, it's a two-step process, and this would be the hypothetical 2 steps involved. So again, in this example, species 1 is defined by a 2n of 6, 3 pairs of chromosomes within the genome. So again, a haploid number of 3. Here's species 2, a 2n of 4, and again, a haploid number of 2. So clearly, in terms of chromosome organization, genome organization for these two species, they're different. Okay. So, but they're closely enough related biologically that they can intermate. Okay. So if that's the case, meaning, when we say able to intermate, the gametes produced by each of these two distinct species have the ability to recognize each other, interact with each other, lead to a fertilization event, and then subsequently in that zygote, that the two haploid sets of chromosomes are able to drive a normal developmental program such that a live birth results. Okay, so this would then represent the viable, the live offspring produced through that interspecific hybridization event. So this individual, again, if you look at the chromosome constitution, it's unusual. So there's 3 copies of A1, or one copy of each for A1, A2, and A3 derived from species 1. So one haploid set of chromosomes. And then the other 2 chromosomes, B1 and B2, derived from species 2. Again, all 5 of these chromosomes are now combined into a single genome. So if this individual wanted to reproduce, it wouldn't have that capability. And again, why would that be? So the reason that that would be is that there's no pairs of chromosomes. Okay, so again, what we know about meiosis, we've talked about from the very beginning, is that you have to have pairing of homologous chromosomes taking place in early prophase 1 to eventually create a bivalent and then a subsequent tetrad. And then it's the formation of that tetrad that allows for the reductional segregation that occurs during anaphase 1, and then the equational segregation that takes place during anaphase 2 to produce functional viable gametes. So in this case, none of these 5 chromosomes have a partner to pair up with. So this individual would be sterile. Okay, so if they're sterile, well, how could you then derive from that sterile individual a fertile, viable allopolyploid derivative? So what it would require is a chromosome doubling event. And what do we know about chromosome doubling events is that they're caused by global non-disjunction. That's actually how we talked about these global non-disjunction events occurring in the generation of an autopolyploid. So it's the same thing here. So if you have an individual who sells this sterile hybrid, all of the cells of the organism consist of these 5 chromosomes. If in some of those cells within reproductive tissues undergo a global non-disjunction event, meaning that you have replication, chromosome replication without segregation, then you produce this chromosome doubling result. And at this point now, this new cell would have 2 copies of each chromosome. And therefore, each chromosome would have a homologous partner to pair up with during early prophase 1 of meiosis. And therefore, you can produce, meiosis can proceed normally, and you can produce functional gametes. And therefore, the organism is able to propagate itself, able to reproduce. So the question is there any evidence that this particular 2-step scheme for producing the allopolyploid, is there actual evidence that this actually occurs? And of course, there is. So let's just look at the first step where you generate the sterile hybrid. So again, in this case, you have to have 2 organisms, 2 species that are biologically closely related. They're different in terms of their genome organizations such that they represent different species, but closely enough biologically that they can intermate and produce a live, viable offspring. So the classic example of that is the mule. Okay, so the mule is an interspecific hybrid between a female horse and a male donkey. So again, the horse has a 2N of 64, a haploid number of 32, and then the donkey, 62 is the diploid number, 31 is the haploid number. So through an interspecific hybridization event, specifically a female horse and a male donkey, you can combine those 32 chromosomes of the horse with the 31 of the donkey to create a mule. But the mule is sterile because those 32 chromosomes are non-homologous relative to the 31 derived from the donkey, so there's no pairing that occurs. Okay, so this mule is sterile. Okay, so what about you can also perform the reciprocal cross, and the reciprocal cross would be a male horse and a female donkey. And in that case, you generate a sterile hybrid offspring. But in this case, instead of calling them mules, they call them hinnies. So there is a subtle distinction, a subtle difference between the two. And my question to you is, why do you think that would be? There's actually several reasons, but based on things that we've discussed, why do you think that might be? Why would a hinny be different from the mule? I mean, is there any genetic difference between the two? So you can look and read this narrative up here in terms of phenotypic differences that distinguish the mule from the hinny. There's differences in the ears, the nature of the way the mane forms, the tail, things like that. So why would there be these subtle differences? Well, first, there's going to be environmental causes, again, because if you have a female horse and a female donkey, the in utero environment is going to be different. So there's the potential, at least, for those sorts of environmental differences to cause the phenotypic differences between the mule and the hinny. But there's another genetic related reason why there would be a distinction as well. And the reason for that is maternal inheritance. So again, remember, the cytoplasm is always transmitted through the female parent. And so in this case, this would contain genetic information within the mitochondria. So in the case of the hinny, the mitochondria are inherited through the donkey, and for the mule, they're inherited through the horse. Okay, so I'll just point out here is that there is a subculture in the U.S. at least, it's probably worldwide, where uh, they race mules as opposed to horses. And so the problem with the mule again is that you can't continue to breed like they do with horses, with the fertile horses, to create newer and better offspring, faster, stronger offspring. So they've actually been able to get around that in the last 20 years or so, in that they've developed the technology where you can actually clone the sterile mules. So again, a form of asexual reproduction for an animal. And so, the cloning procedure is not straightforward, of course, but it's based very similarly on the strategy that they used for creating Dolly, the sheep, the first mammal that was cloned in the late 1990s. Okay, so there's all kinds of examples of these sterile hybrids. So here's another example, a hybrid between a zebra and a horse. And again, you can see clearly, based on the coloration of the fur, is that it's clearly a hybrid where it looks like part horse, part zebra. So again, the animal depicted in this picture here, we would call a zebrula, because the cross involved a male zebra and a female horse. You can actually do the reciprocal cross as well, so you get subtle distinctions, just like I described for the hinny and the mule. And so the reciprocal cross, the outcome would be described as a zorse. Here's another example. You can go on the internet and you can find all kinds of images of these types of interspecific sterile hybrids. So this is a zebra and a donkey. Again, here's the mother, and here's the offspring. So it looks like in this case the father was the zebra. So here's a whole list of other different types of interspecific hybrids, sterile hybrid offspring that are produced through these interspecific hybrids. And one of the classic, again, some of you I'm sure are familiar with ligers and tigons. A male lion and a female tiger produces a liger, or the reciprocal cross produces the tigon. And here's an example of a liger. And again, if you look closely at the animal, exhibits characteristics of both the lion and the tiger. So here's another example. These are pretty dramatic pictures of these animals. They're for ligers, as I said, if you do a search on the internet, they're very large, large animals, and again, a clear and distinct fur coloration hybrid pattern. So here's one other example: a lepon or leopon, a leopard, and a lion. You can create sterile interspecific hybrids of these two animals, these two species as well. And again, the hybrid exhibits characteristics of both original progenitor species. So years ago, there was actually a purported hybrid, a live animal that was a hybrid between a human and a chimpanzee. And I'm sure most of you are familiar with the characterization of the human genome relative to the chimpanzee genome. Again, R2N is 46. For the chimpanzee, it's 48. So they have an extra set of homologous chromosomes in their genome. But at the gene level, at the DNA level, again, very high similarity between the two different species. So this is Oliver. Oliver was a purported hybrid, a human Z, a hybrid between a human and a chimpanzee. And again, he was displayed, as a circus act on television for many years, making lots of money for the owner. So, what were the characteristics of Oliver that would suggest a human chimp, a hybrid? So again, here's an actual picture of Oliver on the right here. So again, clearly characteristics, basic presentation as a chimpanzee, but clearly having human facial characteristics as well. So the other thing that Oliver exhibited that was similar to a human is a much more upright gait and stance, as well as appeared to be much more highly intelligent than your average or your typical chimpanzee. So as I said, Oliver was used to make a lot of money for his owner. So the question is Oliver a true human Z, a true human chimp hybrid? So how would you go about determining that? Well, again, presumably, if that's the case, this would be the sterile hybrid derivative of that interspecific hybridization event. So there should be one haploid set of human chromosomes in the genome and one haploid set of chimpanzee chromosomes in the genome. So that's 23 plus 24, so there should be 47 chromosomes. So of course the owner resisted doing karyotype analysis on Oliver for quite a while, but eventually that analysis was done. And actually what they found is that there were 48 chromosomes in the cells of Oliver. So Oliver is a chimpanzee. Okay, certainly not a hybrid and certainly not a human. So again, it's just, normal variation that exists within populations in terms of, the various characteristics that I described. Oliver was just one of the outliers in terms of, being different from, the rest of the chimpanzee population. So I have Schumann written down there on the slide. So the reciprocal cross. So Oliver would be a human female and a male chimpanzee, and the Schumann would be the opposite, the reciprocal cross. And if you think that there have never been attempts at trying to create this sort of interspecific hybrid, again, I recommend you perform a Google search or an internet search. Okay, so all of those examples of a sterile interspecific hybrid viable hybrid offspring were animals. So there's examples of this in plants as well. I'll just give you two. There's something called a honey bell, a relatively new fruit, maybe 20, 25 years old now. That's actually a hybrid between a tangerine and a grapefruit. So it has, unique characteristics in terms of flavor and texture and so on, that it's been developed into a commercial product now. Still not widely available, but it is available. And so again, one of the advantages of the honey bell, of a fruit being sterile, is, again, we've talked about this previously, like the banana, the odd numbered autopolyploid, is that there's no seeds. So again, that's a beneficial characteristic when you're trying to develop an edible or a fruit as a food source for humans. So here's another example. The boysenberry is actually interspecific hybridization between three different species: the blackberry, the raspberry, and the loganberry. Three different haploid genomes all combined into one new genome to create, the sterile boysenberry, and again, a unique type of berry in terms of texture and flavor, nutrition, and so on. But again, it's seedless because of the lack of the ability to produce viable gametes. Okay, so the question is, if there's all kinds of examples in nature of creating these sterile interspecific hybrids, how do you demonstrate these proposed chromosome doubling events that would restore fertility and actually create a new species, a new allopolyploid derivative? Okay, so again, there are many examples of that. So here is probably one of the better, it's a textbook example. This is in the genus Brassica, again, a plant. So actually what you have is, within the genus Brassica, you have 6 different species that are represented here: the Brassica oleracea, carinata, Brassica nigra, Juncea, campestris, and Brassica napus as well. And all of these different species, you can see, are sources for important food products or food crops for human consumption, particularly among the oleracea. The cauliflower, Brussels sprouts, or they're down here, broccoli, canola is an oil crop, and so on. So, the cabbage also falls within the oleracea. Brassica napus, again, rapeseed, or this is canola within the Brassica napus. You have different mustards that are derived from these other Brassica species, and then, turnips and Chinese cabbage as well. So clearly an important plant genus and important plant species. So there's been a lot of genetic work that's been done on these different species. And actually what they've been able to determine through chromosome analysis of these 6 different species is these 3, the carinata, the napus, and the Juncea, are all allopolyploid derivatives of 3 original diploid progenitor species, which are the Brassica nigra, the campestris, and the oleracea. So how do you determine that? Well, again, you do chromosome counts. So just to give you an example, if you were to take the oleracea, 2n of 18, so the haploid is 9. The campestris, a 2n of 20, so the haploid is 10. You combine them to create a sterile derivative of which the cells contain 19 chromosomes. And so what happened, or what you would then require, is that chromosome doubling event through a global non-disjunction within the cells, the reproductive tissues of this sterile hybrid. If that's the case, then you restore fertility, and the individual can then propagate itself as a new fertile allopolyploid derivative of the original two progenitor species, the Campestris and the Oleracea. And if that were the case, then how many chromosomes would you expect to see within the cells of the plants that fall within the Napus species? There should, you double the 19, there should be 38, and that's exactly what you see. So it's the same thing for the Carinata. The Carinata is an allopolyploid derivative of Oleracea and Nigra. So again, a haploid of 8 here and a haploid of 9. So the sterile hybrid would, the cells would consist of 17 unpaired chromosomes. You get a global non-disjunction event. You double that, which is 34, and that's the chromosome count of these different plants that fall within the Carinata species. And then the last one is the Ginsea, a haploid of 8 from Brassica nigra, a haploid of 10 from Campestris. Combine that, that's 18 in the hybrid derivative, the sterile derivative. And then you get a chromosome doubling to 36, which would restore fertility and create the new species, the new allopolyploid derivative, which is the Brassica ginsea. So what this means is that what happens is, in the wild, without human, all of this occurred totally randomly and totally naturally. So without human intervention. So these plants, the three original species, the Campestris, the Nigra, and the Oleracea, were able to cross-fertilize, cross-hybridize to create those sterile derivatives fairly frequently. And then in a few of those sterile derivatives, there was, again, a random global non-disjunction event that doubled chromosome counts and restored fertility, which then allowed for propagation, reproduction, and also in doing so, restoring that fertility through the chromosome doubling. If you created a new species, you've created a new genome that represents the two diploid genomes of the original progenitor species now combined into one new genome. So all of this occurred totally randomly and spontaneously in nature. So here's another example of this as well, although in this case, there was a little bit of human intervention. So about 10,000 years ago, humans settled from, a nomadic hunter-gatherer type of existence to settling down into small communities and, started the process of farming, growing food instead of hunting and searching for food. So, of course, what that required was retaining seeds from one year to the next, so that instead of eating the seeds, you would save them, plant them the next year to grow next year's crop, next year's food source. So this was done among different wheat varieties or species, actually. Okay. So again, initially there was little, if any, human intervention in terms of the generation of modern day wheat, which is an allopolyploid derivative of original diploid species of wheat. But eventually what happened is that, again, because of the benefits conferred by the allopolyploidy, humans selected for that form of wheat such that that is what is predominantly grown nowadays and consumed by humans in the present day. So this is how the process went. So again, totally randomly, totally spontaneously, two different diploid forms of wheat. This is einkorn wheat. It consists of what wheat geneticists call the A genome, so a diploid species. And then you had the Triticum circe, which is a different species of wheat that contains what we geneticists call the B genome. So these two closely related species could cross-hybridize and generate sterile plants, sterile progeny plants. And again, among some of those plants, there was a random spontaneous chromosome doubling event that restored fertility and created a new species. So in this case, the interspecific hybridization between Triticum monococcum and Triticum circe produced this allopolyploid derivative, more specifically an allotetraploid, Triticum turgidum, which now contained a diploid copy of the A genome and a diploid copy of the B genome. So then what happened, again, totally randomly and totally spontaneously, is that this allotetraploid, remember, tetraploids, just like with the auto tetraploids, if you were an auto tetraploid, is that you're fertile. So these individuals in this new species, the turgidum, are fertile as well. So again, what happened is that there was an interspecific hybridization with a third species of wheat that contains what wheat geneticists call the D genome. A sterile hybrid many were produced, within which a few of them, there was a chromosome doubling event in which, fertility was restored and allowed for propagation as a new species. In this case, this would be an allohexaploid, because you've now added, or you've combined 3 diploid genomes into a single new genome. So this actually represents modern day wheat, bread wheat. It's a hexaploid, it's an allohexaploid, consisting of these 3 original diploid genomes of those 3 original progenitor species. So you say, well, how is this the particular form of wheat that was maintained by humans over the last 10,000 years or so, such that we still use it nowadays? Well, it's the idea, again, the same as we talked about with autopolyploids, is that as you increase chromosome number in the cells get bigger, and therefore all of the components, the structures, the tissues, the organs of the plant get bigger as well. So in other words, the grains of wheat that are found on this allohexaploid are bigger than you would find on any of these original diploid progenitor species, or even the tetraploid, such that would be the one that the human populations would maintain across, stored during the winter, so they could grow out next year's crop that would then ensure a high yield or a large harvest to feed the community. So again, these are all spontaneous events. In this case, again, it involved a little bit of selection on the part of humans as well. But the question is can you also generate these types of allopolyploids artificially? Well, of course you can. Just like we saw that you can generate the auto polyploids artificially as well with colchicine. So this was first attempted in the early 1900s, the 1920s, by this individual, a guy by the name of Karpachenko. So what he wanted to do, his idea was he wanted to combine the characteristics of Brassica oleracea. Again, remember, that's where broccoli, cauliflower, Brussels sprouts, and other vegetable crops are derived. Are combined those characteristics with the radish. So this is the species within the genus Raphanus. So what he would do is he would artificially, just like Mendel did, cross-hybridize the brassica with the radish. And, he may have performed, say, 100 crosses or more, and he might have obtained maybe one viable seed. because, the biological differences, the fertility level is going to be very low in this type of interspecific hybridization event. So, but in any event, he got one viable seed. So he grew it out, he got a plant. However, that plant was sterile. Again, it contained 9 chromosomes from the radish and 9 from, say, broccoli. Okay, so a sterile hybrid. So what did he do? Well, he treated the reproductive tissues of that hybrid plant with colchicine to force a chromosome doubling event, or increase the frequency of non-disjunction events, the chromosome doubling. And in doing so, he then restored fertility. And so he was able to then propagate that fertile plant and generate this new allopolyploid or allotetraploid derivative that he called Raffinose brassica. And again, the Raffinose brassica now consisted of a total of 36 chromosomes, the diploid genome of 18 from radish and the diploid genome of 18 from Brassica oleracea. So the problem was what he was hoping for, again, by combining the characteristics of the 2 original species, he was hoping to create a plant that, the above ground portion of the plant would produce a head of broccoli, and the below ground portion of the plant would produce a radish. Okay, that was his goal. But unfortunately, he got the exact opposite outcome. In other words, the above ground portion of the plant looked like the radish, so pretty much just a bunch of leaves, and then the below ground portion of the plant looked like broccoli, so just a root system. Okay, but in any event, what he was able to demonstrate through this experiment is that you can artificially create these allopolyploids. So subsequent to that, there have been some successes where they have artificially created these types of allotetraploids that are now grown to produce food for human consumption. And I'll just give you one example of that. Some of you may have heard of this grain that we call triticale. And triticale is actually an interspecific hybrid between wheat and rye. So actually a tetraploid, an autotetraploid wheat with a diploid rye. And so you end up with, combining those 2 genomes, the 4N of wheat with the 2N of rye, to give you a 6N triticale derivative, the allopolyploid derivative. So how did they do it? They did it the same way that Karpachenko did, using their tweezers, produce many interspecific crosses between the 2 different species, the wheat and the rye. You get maybe 1 or 2 viable seeds. You grow those seeds up. They're sterile. You treat the tissues with colchicine. You get a chromosome doubling, restore fertility within the flowers of the plant. And then in doing so, you've created a new species. You've created, in this case, the triticale. And so triticale is not, it's not a major food crop just yet, but it is, grown to a fairly large extent in some South American countries and European countries, as indicated here, Poland, Germany, and France. And if you're familiar with these Kashi food products, is that they're some of them are produced from a 7 different grain combination. Well, one of those 7 grains is, if you look at the ingredients, is triticale. Okay, so the question is if you have, say going back here, if you have, say, Brassica napus, and you suspect that Brassica napus is an allopolyploid, how would you confirm that experimentally? Or, what sort of genetic test would you perform in order to identify the 2 original progenitor species for Brassica napus? So this is actually what they did, is that what you would do is you would hybridize the proposed or the suspected allopolyploid with what would be presumed to be one of the original progenitor species. So here, again, if you cross, this is the allotetraploid here, AABB. If you cross that with a presumed progenitor species that has a diploid genome of, an AA set of chromosomes, then what you would expect is to generate a sterile hybrid derivative. And then what you would do is you would look at reproductive tissues within that sterile hybrid, look within the flowers for cells that are undergoing meiosis, and you would look for pairing of chromosomes. So in this case, this A set of chromosomes derived from this presumed progenitor species would be able to pair with the A genome derived from this allotetraploid. So you get a pairing of 9 of the 18 chromosomes. You'd see 9 tetrads at early prophase 1 of meiosis. And then you'd also have 9 unpaired chromosomes. So if you see that sort of configuration in the hybrid, then you know that this individual, with the AA diploid genome is an original progenitor species of the allopolyploid. And you could do the same thing with the second progenitor species, presumed progenitor species that now contains the B set of chromosomes. So here's your suspected allotetraploid. Here's a suspected progenitor species. You generate a sterile hybrid. You then look at reproductive tissues. You look within the flowers for cells that would be, trying to undergo a meiotic event. You capture cells at early prophase 1 of meiosis. And if you see any chromosome pairing, in other words, any tetrads, then you know that this individual was a progenitor species for this allotetraploid here. If you see no pairing, so in other words, if you saw a total of 27 chromosomes in the hybrid, but there was no pairing, no tetrads forming, then that plant would not be a progenitor species. Okay, so one other example, or two other examples I want to show you. So again, for these allopolyploids, the examples I've given you are in plants. And again, that's where you see it most frequently, because when you create an allopolyploid, as I've said before, it generates a genetic imbalance. You have too much genetic information. You're deviating from the normal diploid state. And so that imbalance can be tolerated in plants, but not very well in animals. So it's very rare that you see an allopolyploid animal species. But there are a few examples. And so here's one. This is a type of tree frog. So there are two diploid species, closely related species of this particular tree frog, an eastern population, with this range here, and a western population over here. Well, they intercrossed, or they intermated to create, again, the original sterile diploid, or the sterile derivative. There was a chromosome doubling event in one or a few of those hybrid individuals to restore fertility and create an allotetraploid derivative of those two original diploid species. And this is a tetraploid species of tree frog that has this now a third range, more in the southern portions or southern states of the United States. And here's one other example, and this is actually a recent determination through more modern genetic analysis. So there's a type of frog, Xenopus laevis, that has been used as a model system for a variety of different types of genetic studies, usually relating to questions about development, genetic control of development. So of course, because of the importance as a model system for genetic analysis, you want to understand the genetics of the organism very well. And what they've recently determined is there's pretty strong evidence that this particular species of frog, the clawed frog, is an allotetraploid of two original progenitor diploid species. So one of those species is extinct, but the other one is still alive, or still exists. So this is what they figured out: is that Xenopus has a 2n of 36, and the viable or the still living related species, the Tropicalis, has a 2n of 20. Okay, and then the extinct species has a 2n of 18. So you take what they believe happened is that there was an interspecific hybridization of the two progenitor species, which then, after a subsequent chromosome doubling event, created the now modern day version or species of the western clawed frog that is Xenopus laevis, this new species. And what they're showing you here in this diagram is this is one of the original progenitor species. This is the Xenopus laevis, and you can see it's almost twice the size of the diploid species. So again, it's the idea that as you increase chromosome numbers within the genome of the organism, in this case through allopolyploidy, is that you increase the size of the organism as well. So that seems to be clearly evident in this case. Okay, so I'll wrap up this discussion with this example, and then next time we'll be talking about structural chromosomal abnormalities in contrast to what we've been doing, which is number variants for chromosomes within the genome, the aneuploids and the polyploids.